Picture this. 4 billion years ago, Earth is a chaotic, churning mess. And somehow, out of that primordial soup, the very first sparks of life ignite. But how? I mean, that's one of the biggest questions in all of science. Today, we're going to dig into a really crucial piece of that puzzle, how the very first container for life might have actually formed. So just think about it for a second. All life that we know, everything, happens inside cells. They're these amazing little packages that keep all the molecular machinery organized and working. But if you rewind the clock, way back before the very first cell existed, how did the essential ingredients for life even find each other to get the chemical party started? Well, the answer is you need a container. Scientists have a term for this. They call it a prebiotic compartment. But really, it's just a tiny, protected bubble. It's a special space where all those key molecules could get concentrated, start bumping into each other, and do the complex chemistry needed for life instead of just floating away in a huge primordial ocean. So the hunt is on. We're looking for a simple, plausible candidate for this very first container. What could have just popped into existence on a young, messy Earth and acted as life's first little chemistry lab? And this idea of concentration is absolutely crucial. I mean, imagine you're trying to bake a cake, but instead of using a bowl, you just sprinkle the flour, sugar, and eggs into an Olympic-sized swimming pool. It would never, ever work, right? The ingredients are way too diluted. It's the exact same problem for the origin of life. You have to bring the building blocks together in one place. And that brings us to a really fascinating candidate that has scientists super excited. And it's these simple, membraneless droplets made of something called polyesters. So, what on earth are these things? Now, these are nothing like the complex, sophisticated membranes in our cells today. They are much, much simpler. You know how oil and vinegar separate in salad dressing? These polyester microdroplets do basically the same thing. They're tiny, oily blobs that naturally separate themselves from water, creating a nice, self-contained little environment. And what's so brilliant is how they could have formed. Just imagine early Earth with all these ponds and hot springs constantly getting wet, then drying out over and over. You'd have these simple organic molecules called α-hydroxy acids just dissolving in the water. Then the sun beats down, the water evaporates, and the heat essentially bakes these molecules together into long chains, polyesters. The next time it rains and the pond fills up, boom, these polyesters just spontaneously assemble themselves into droplets. It's an incredibly simple and plausible mechanism that doesn't require any magic at all. But hey, it's a nice idea, but could it really happen? To find out, scientists had to put these droplets to the test. And first up was a big one. Could they handle the extreme temperatures of early Earth? And you know, this is a real make-or-break question. Early Earth was a wild place. You had environments that were freezing cold, and others that were literally boiling hot, right next to volcanoes. So for these droplets to be a serious contender for life's first bubble, they couldn't be too picky. They had to be able to form under these crazy conditions, especially in those hot geothermal spots where this chemistry was most likely to happen. Okay, and here is the proof. And honestly, it is stunning. Take a look at the image on the left. This was created at a pretty cool 40 degrees Celsius. You can barely see anything, right? But now look at the image on the right. This experiment was run at a much hotter 80 degrees, a totally realistic temperature for a geothermal pond back then. All of a sudden, the solution is just packed with these perfect, distinct droplets. The difference is just night and day. So, what is going on here? Why does that extra heat make such a huge difference? Well, this chart breaks it down perfectly. The heat is what provides the energy to link all those simple molecules into long polyester chains. At the lower temps, you only get these short, stubby little chains. But crank the heat up to 80 degrees or more, and you get much, much longer ones. And those long chains are more hydrophobic. They basically hate water, just like oil does. And that water-hating quality is exactly what makes them clump together so well to form stable droplets. So the verdict from this first test is crystal clear. These little cradles of life don't just survive the heat, they actually thrive in it. Hotter environments like the edges of volcanoes or geothermal springs would have been the perfect factories for churning out these essential bubbles. All right, so the droplets passed the temperature test with flying colors. Awesome. But now they're up against an even stranger, maybe even more profound challenge from the origin of life, the puzzle of molecular handedness. So this concept is called chirality, and it's pretty simple, really. Just look at your hands. They're mirror images of each other, right? but you can't perfectly stack your left hand on top of your right. Well, a lot of life's molecules are just like that. They come in left and right-handed versions. The really weird thing is that all life on Earth today pretty much exclusively uses one version, like only left-handed amino acids. This brings up a huge question for scientists. The primordial soup, it wouldn't have been neat and tidy with all the left-handed molecules over here and the right-handed ones over there. No way. It would have been a messy 50-50 mix of both. So did these first containers need pure, pre-sorted ingredients to form? Because if they did, that's a huge problem. Or could they just work with the messy, mixed-up stuff that was actually available? So, the researchers tested this head-on. They tried making the droplets using only left-handed starter molecules, then only right-handed ones, and then a messy mix of both. And the result was, honestly, it was incredible. It made almost no difference at all. The polymers that formed were the same length, and the droplets that came together were pretty much identical. And you can see the results for yourself in these images. It just doesn't matter if the ingredients were pure left-handed, pure right-handed, or that messy mix of both. In every single case, you get these fantastic little droplets. This is huge. It means the very first step, just making the container, didn't need to solve this complicated handedness problem first. It just worked, using whatever messy ingredients were lying around. And let's be clear, this is a really big deal for origins of life research. It completely removes a major hurdle. The very first bubbles didn't need to be picky. They could form from the chaotic, mixed-up chemical junk of early Earth, which makes them a much, much more robust and plausible candidate. So, after passing both of these crucial tests, the heat test and the handedness test, what does this all mean for the big picture of how life got started? Let's put all the pieces together. So, what's the big takeaway here? It's that these simple polyester droplets are surprisingly tough. They are not delicate, they are not demanding, they actually thrive in the hot, messy conditions we think existed on early Earth. And they can be built from the mixed-up, chaotic ingredients that were available. And that combination of toughness and simplicity makes them a really powerful candidate for life's very first bubble. And you don't have to take my word for it. The scientists who did this work concluded, and I'm quoting them here, that the wide variety of conditions these droplets can form under enables many possibilities for such systems to have played a role during the origins of life. For scientists, that is a very, very exciting conclusion. And that leaves us with a really powerful final thought. Isn't it incredible to imagine those turbulent geothermal ponds on a young Earth, not as sterile, empty pools, but as bustling nurseries filled with trillions of these simple little bubbles, each one a tiny potential experiment, setting the stage for the incredible journey of life to finally begin.